So yeah, thank you again, everybody, for coming to listen to my talk, and thank you for this award. So I'd like to tell you today about the research I've been doing as part of this postdoc project, using computational approaches to investigate and identify mechanisms of structural cardiotoxicity. But before diving into that, I'd like to just first give an overview of how we view cardiovascular safety in AstraZeneca. So we have different aspects which we consider. One of these, of course, is electrocardiograms. So generally, we have a very good understanding of the molecular mechanisms underlying ECGs, how these translate between in vivo or in vitro models. And also, we understand what clinical biomarkers we can use to interpret these ECG intervals and waveforms. For hemodynamics, we also have a fairly good picture of this as well. But for other aspects of cardiovascular safety, like structural cardiotoxicity and also inflammation in the myocardium, the picture gets a little bit fuzzy. First of all, we don't really understand the molecular mechanisms that drive these outcomes. And also, we currently presume that there is some species translation, but we don't really have a lot of evidence to support that. And finally, if we consider biomarkers, this is really a work in progress around trying to identify additional biomarkers, but also validate them for use in cardiotox assessment. Today, I'll be focusing on talking about structural cardiotoxicity. But first of all, what even is structural cardiotoxicity? In the clinic, structural cardiotoxicity is defined as changes in left ventricular ejection fraction, or else the ability of your heart to pump blood to the rest of your body and back. And this can also lead to changes in structure in cardiac tissue, and also to heart failure, and in very severe cases, death. So this is an outcome that we want to avoid at all costs in early drug discovery. In early drug discovery, we have some phenotypic assays to assess SCT. So the one that we commonly use in AstraZeneca is called the high content biology imaging assay. So in this assay, we assess 3 different parameters. The first is mitochondrial membrane potential. The second is endoplasmic reticulum integrity. And the third is ATP depletion, or a measure of cell viability. We often use this assay, but we are exploring other assays which could give us insights into other aspects of structural cardiotoxicity. One of these is the calcium transience assay. So calcium signaling is important for contractility in cardiomyocytes. And what you're looking at here is an example of a calcium transient trace. So we have time in seconds on the x-axis and calcium intensity across cardiomyocytes on the y-axis. And what you can see is that over time, the calcium abundance will increase as the cardiomyocytes contract and then decrease as they relax. And this continues in a cyclical form. So if we extended the time axis, we would see the cycle repeating itself. Recently, we had a collaborator who developed an algorithm to calculate different parameters from this calcium transient trace. So one of the aspects of my project was to try and understand whether we could use this assay to identify additional parameters for SCT assessment. So that's one part of what I'll be covering today. But another challenge we face in structural cardiotoxicity is that even when we have these different assays at our disposal, it's very difficult to link them to the molecular mechanisms which drive SCT. Therefore, when we identify a drug which is potentially structurally cardiotoxic, more often than not, we will shelve that compound, and that leads to a lot of wasted time and money in trying to develop that compound in the first place. So if we can identify molecular insights, then we could potentially de-risk our compounds and save some of these compounds for future development. So this leads us to what we'll cover today. So first, I'll talk about the calcium transient assay and how I identify potential assay parameters which could be used to study structural cardiotoxicity. And then afterwards, I'll go on to talk about the molecular mechanism aspect of it. So this includes looking at transcriptomic data, but also looking at metabolomic data to try and enhance our understanding of structural cardiotoxicity. So before we start working on our different assays, we first need to identify a physiologically relevant cell model. Here we turn to a cell model called human-induced pluripotent stem cell-derived cardiomyocytes. These are cardiomyocytes which beat under the microscope, so these are perfect for our contractility assay. And so for this experiment, we added our compounds of interest over an 8-point concentration response, and we incubated our cells for 72 hours. After that, we add a fluorescent calcium dye, and we put this into a FLIPR tetra machine, which then measures the calcium abundance in 800 reads across 3 minutes. This will give us the calcium transient trace that we saw in the previous slide. And as I mentioned, using our collaborators' software, we were able to identify 24 different parameters. Just to try and demystify the process a little bit, how this algorithm works is that it detects some peaks of some points of interest in the trace. This red point, for example, is a peak. That's the highest point of the waveform in a given time frame or the given cycle. We have these orange points as well at the bottom, which mark the starts of the cycles. And we also have these green points, which mark where the algorithm thinks the cardiomyocytes start to relax, also known as the tail. So then from that, from these reference points, we can calculate our parameters of interest. For example, the peak space is the time between two adjacent peaks. We can have the tail duration as well. That is the time it takes to go from that green point to the orange point, and so on. But with this many parameters, we also need to whittle them down and see which of these are important for structural cardiotoxicity. So for this, we turn to machine learning methods as a way of selecting the features of interest. At first, we include all 24 parameters in our machine learning models, and we don't really know how these parameters might interact with each other and with structural cardiotoxicity. Maybe they have additive or linear effects. So for this, elastic net and ridge regression are very good models for modeling those linear effects, but equally they could have nonlinear effects. So for this, we use a random forest model to try and capture those potential relationships. After that, we look at the important features of our different models, and we identify our handful of selected features. But even so, we need another way of benchmarking how well these features would perform in classifying SCT. I didn't mention this before, but for calcium transient assays, we usually use peak count, that is the number of red points in our trace, and average amplitude, which is essentially the height of a given peak, as gold standard parameters. So to try and benchmark the features we identified, we generated a logistic regression model of our selected features and compared that with an equivalent that includes only peak count and average amplitude. So I call that the baseline model. And now we come on to the results. So first of all, what kinds of compounds did we use? We used a variety of compounds which were chemically diverse, and we focused particularly on a marketed or commercial set of compounds using the FDA labels for using their FDA labels to classify them as structural cardiotoxins or non-structural cardiotoxins. Given that we had such a small data set, we used the marketed compounds both for model training and model testing. This is suboptimal from a data science perspective because ideally you want to have a model that you can test on an independent set and show that it's generalizable. But given the data set we had, we simply used the marketed set for model training and testing. Still, we wanted to try and preserve that independent testing element. So we also included a set of preclinical compounds, which were labeled for SCT based on in vivo data. And we therefore apply our machine learning models onto this set and try and see how well they perform on the preclinical set. So now we come to the model performances on the left-hand side, and perhaps what you're thinking is this doesn't look very impressive, and I'd be inclined to agree with you on that. So if we look at the cross-validation performances, you can see this varies quite widely across the sets of compounds we use. And even though this performs slightly better in the marketed set as a test set, it doesn't perform very well in the preclinical test set. In some ways, this is expected. The reason is because calcium transience or contractility is only one aspect of structural cardiotoxicity. Therefore, it only represents one facet, and therefore we can't really expect it to be an all-encompassing measure of structural cardiotoxicity. But furthermore, the aim of this project, the aim of this experiment wasn't to build a brilliant predictive model of SCT, but rather to identify some parameters which could be followed up as potentially important for SCT assessment. So what I did here was I measured the feature importances in each of the different machine learning models, and then prioritized those which were highlighted in at least 2 of them. And what I got after that was a set of 9 parameters, which were highlighted in at least 2 of the machine learning models as important features for SCT. These are summarized in this trace here. So we have peak count as a usual suspect, but we also have different parameters like the width of the peak at different heights of that peak. We also have something called average shoulder amplitude. It's not very obvious in this model trace, but sometimes you can get a peak which is a little bit shorter than this main peak and very close to it. So we call that the shoulder. So average shoulder amplitude came out as an important parameter as well. We also had average tail duration, average inner lambda, which is like the period for a cycle, the average valley, which is the low point in the cycle, and also the maximum and minimum intensity. So finally, we took these 9 different parameters and we put them into a logistic regression classifier and compared them to a baseline classifier of SCT. The results here are encouraging. So with the cross-validation and commercial test performances, we can see that the 9-parameter model performs better than our baseline model, although the performance is moderate for the preclinical test set once again. So taking all of these results together, we can say that we identified some additional parameters in the calcium transient assay, which could capture mechanisms of SCT relating to contractility. These parameters could therefore complement other in vivo and in vitro approaches to assess SCT, like the high-content biology imaging assay, which I mentioned earlier. However, of course, the caveat is that our predictive model had a moderate performance, and also that we have a very small compound set. Therefore, future work would be required to validate the identified parameters for SCT assessment. But now we come on to the other aspect of what I mentioned in my introduction, which is that even when we have these additional parameters, we still need to make them interpretable somehow from a molecular mechanistic perspective. And this is where we now move on to the second part of my talk, which is about identifying potential mechanisms of SCT using transcriptomics and metabolomics data. So once again, we come back to our physiologically relevant model of interest. And instead of using cardiomyocytes in this study, we use something called cardiac microtissues. We're still using our human-induced stem cell-derived cardiomyocytes here, but in addition, we also add endothelial cells and fibroblasts to our mix. So essentially, we culture these different cell types together for over 14 days, and at the end of our 14 days, we have a cardiac microtissue that is a ball of cells, which when we look under the microscope, can also see that they're beating. So for this study, we expose our cardiac microtissues to different compounds of interest. In this case, we used a smaller set of structural and non-structural cardiotoxins because we wanted to make sure they were chemically diverse, but also had very clean sort of profiles of structural cardiotoxicity and non-structural cardiotoxicity. So we exposed them across 3 different time points and 2 concentrations, and we perform metabolomic and transcriptomic profiling on them. But first, I'll talk about the transcriptomic aspect of this study, since the transcriptomic analysis is more mature at the moment. So with omics workflows, it's always important to make sure you have a bioinformatic pipeline in place. First, of course, you have to pre-process your data set and make sure that, that the quality of it is good. So some of the considerations we would typically have for a transcriptomic data set include batch adjustment. In our experiment, we performed the profiling in different batches. Therefore, we need to normalize across the batch effects to make sure that doesn't affect our results. But we also have to normalize, for example, for different library sizes when we're looking across different samples, or sometimes genes which have, longer transcripts tend to be more easily detectable than shorter transcripts. So again, we have to normalize for the transcript length or reads. After all this normalization is done, we typically have a data visualization step. We want to make sure that our data looks the way that we expect it to, but we also want to identify any potential covariates or confounders which either we want to adjust away the effects of or just be aware of when we go through our statistical analysis. After the data visualization step, we then have our statistical pipeline. And I'll go into each of the different methods that I've listed here, but just so that you have an overview for now. We perform 2 complementary methods, one called differential expression analysis and another called co-expression clustering analysis, to identify genes of interest from our data set. Afterwards, we perform machine learning to, again, with a feature selection step to identify a smaller set of important genes, and this will give us our prioritized gene expression signature of SCT. So before this slide, I've done the batch adjustment, I've done the normalizations. What you can see here are the plots colored by different covariates. So experiment is our batch effect. So since I've already performed the batch adjustment, we can see that the different clusters, the different experiments don't really cluster together, and that's a good sign. If we turn our attention to the compound plot, we can see that some of the samples cluster together by compound. So one example here is fluorouracil. We also have idarubicin on the right-hand side. And on the left-hand side, we can see that sorafenib and sunitinib samples tend to cluster together as well. If we look at time point, we can also see that there's an effect there. So we can see that there's a 6-hour time point, which is very different from the 48 and 72-hour time points. And finally, if we look at our structural cardiotoxicity labels for the samples, we actually see it's a bit of a mess. There's not really any clear clustering. But what this made us think, what this set of plots made us think, is that the effects that we're seeing primarily belong to the compound data set. So maybe the best way of analysis first is to do a compound versus control analysis, identify the genes which are differentially expressed in those conditions, and then cluster our compounds by similarity of gene expression profile, and then identify potential signals of structural cardiotoxicity that way. Unfortunately, it wasn't quite as clear-cut as that. So what you can see here is a heat map. And for this analysis, we only took the treatments which gave us the largest number of differentially expressed genes for each compound. So these are the treatments that you see along the columns in our heat map. In the rows, we have about 4,000 genes, which were significantly differentially expressed in at least one of these treatments compared to control. But when we do the clustering and we just sort of draw a line to identify 4 different clusters, we can see actually the compounds don't really cluster by structural cardiotoxicity. Furthermore, when we looked at the differentially expressed gene groups, which came up in each of these clusters, the enrichment analysis didn't really tell us anything useful about SCT. So in the end, this approach didn't really work very well for us. Therefore, we decided to go down, we decided to use other approaches. So one of these is differential expression analysis, but instead of looking at compound versus control, this time we look at structural cardiotoxins versus non-structural cardiotoxins. And again, because each of our different compounds could have maximal effects at different concentrations or time points, we compared the treatments based on the number of, we only used treatments which had the largest number of differentially expressed genes for each compound. So the question here we're asking is essentially which of our genes are differentially expressed in structural cardiotoxins compared to non-structural cardiotoxins? And in our data set, we have 18,000 genes. So that's quite a lot of genes to be assessing. And maybe you wouldn't be surprised then to find that 2,600 of these are significantly differentially expressed at a false discovery rate adjusted p-value of less than 0.05. But in transcriptomic analysis, and especially in a data set with very small sample sizes like ours, it's very easy to get a lot of noise and false positives. So we wanted to try and filter down this set of initial genes to get a set of genes which we were confident in. So first we applied an even more stringent, another filtering criterion, which was to have the absolute log2 fold change larger than 1. When we did this, 59 of our genes were still significant. And then we applied a few more different methods of filtering down the set of 59 genes. The first method we took was to make this log2 fold change threshold even larger. So when we subset to 1.5, we then had fewer genes which came out as with large fold change effects. We also applied a partial least squares discriminant analysis to the data set to see which of our 59 genes were important for discriminating between toxins and non-toxins. And partial least squares discriminant analysis is essentially a slightly different, a variant of PCA. It's a bit of an oversimplification, but as just to keep it simple. But we also validated the genes that we found in an independent transcriptomics data set. So when we applied these 3 different approaches or filtering criteria and put together the union of genes, we found that 17 protein coding genes of our 59 were prioritized for follow-up and were robustly associated with SCT. So that was the differential expression analysis. But as I mentioned, differential expression analysis can be very sensitive to small sample sizes. So we wanted to try a complementary analysis method to build confidence in the set of genes that we identify from this data set. For this, we turned to a method called weighted gene correlation network analysis, or WGCNA. The premise of WGCNA is that it clusters the genes in the data set into different clusters or modules based on how correlated their gene expression changes are with each other. So the premise is that if you have 2 genes which are upregulated together or downregulated together, or changing similarly for that matter, then they tend to be representative of a similar functional pathway. So ideally, each of these modules would represent different functional pathways of SCT. But of course, because we're looking at different compounds, these modules could also represent mechanisms of action for the compound and other things besides SCT. So we need to correlate these modules with SCT and also with SCT-related assay parameters. So again, we bring in the high content biology imaging assay parameters, as well as the calcium transient assay parameters, which I prioritized previously. But when we identify these modules, we identify them at the module level, but we're interested in the gene level. So what we ideally want to do is look at our modules and identify genes which are important within the modules. We call these hub genes. And hub genes can be defined in many ways, which I'll go into later. But essentially, we want to prioritize the hub genes in SCT correlated modules and prioritize them as genes which could be linked to structural cardiotoxicity. So in our analysis for WGCNA, we identified 23 different modules altogether in the data set, but only 5 of these were correlated with SCT. WGCNA assigns different color names to each of the modules, so that's why you see color names in the module column in this table. But across these modules, there are 2 different patterns of correlation. The first cluster of modules we see are with the green, dark gray, and orange modules. These were positively correlated with structural cardiotoxicity and also with high content biology imaging parameters, but negatively correlated with calcium transient assay parameters. And when we perform an enrichment analysis on the genes in each of these modules, we get pathways linked to mitochondrial dysfunction and contractility. Conversely, we have a second cluster of magenta and black modules, which had inverse correlations with SCT and HCB parameters, but were positively correlated with calcium transient assay parameters. And when we look at the enrichment analysis for these, we can see that the genes are enriched in pathways linked to extracellular matrix organization, focal adhesion, and activity of compounds involved in cell integrity and structure. So these results are promising because in the literature, these same pathways have been implicated in cardiotoxicity linked to known structural cardiotoxins. So now we come on to the hub gene identification step for each of these modules. In this study, we have 3 different ways of defining a hub gene, or 2 different ways rather. So one of them is by looking at a metric called module membership. Essentially, this is correlating the expression of a given gene to the first principal component of all the genes in a module. But equally, you could say, why do we need to look at the first principal component? We could just look at the correlation of a gene with all of the other genes in a module and see how correlated that gene is. And the more correlated it is with more of the genes, the more connected it is. So this is our second measure, our second definition of a hub gene, which is intramodular connectivity. But because we are also interested in hub genes in the context of SCT, we also applied a third criterion here, which is that our genes need to be associated with SCT as well. So when we apply these 3 metrics across the different modules correlated with SCT, we identified 356 hub genes. Finally, if we bring these together with the 17 prioritized genes from the differential expression analysis, this gives us a total of 367 unique genes which are linked to SCT. Now, at this stage, we could very well have dusted our hands off and said that, you know, we've got our 367 genes. These are all very interesting to follow up, and an end of story. But the reality isn't quite like that, because in our particular use case, we want to be able to profile a panel of genes for SCT assessment, and 367 is just too many genes. And that's why we then apply a machine learning approach to select important features from these 367 genes. So as a reminder, here is our data set. We have 144 samples relating to our 12 different compounds of different time points and concentrations. And here we include 328 genes. So these are the 367 minus any genes which had low variance in the data set or genes which were highly correlated with each other. So we got rid of one of any pair of highly correlated genes. Then we built a range of machine learning models. So again, we were thinking about linear and nonlinear effects. And because we have a small data set, once again, we used relatively simple machine learning methods. So here we use logistic regression, elastic net, and random forest. And this time, because we were interested in the performance of our models, we then benchmarked these against something called a dummy classifier. The dummy classifier is a very crude classifier. So it essentially looks at the balance of classes in our data set. That is 67% toxins, 33% non-toxins. So when you give it a new sample to predict, 67% of the time, it will say it's a structural cardiotoxin. We then assess the model performance, select the most important parameters, and then identify our pool of important genes, which could be linked to SCT. Just as a side note, I also wanted to mention that cross-validation is important for machine learning. I won't go into the details for this particular slide, but the main takeaway from this is that because we had multiple samples linked to the same compound, it was very important to make sure that samples linked to the same compound didn't turn up in both the training sets and the validation and test sets. So this is just a schematic showing how exactly we divided the toxins and the non-toxins across our different data sets. And now we come on to our results. So if we look at the box and whisker plots on the right-hand side, we have three different ways of assessing our models. One is the area under the curve. One is the precision, that is how well of all the structural cardiotoxins you predict, how many of them are actually structural cardiotoxins, and the recall, which is of the true cardiotoxins in the data set, how many of these do you predict correctly? So with our dummy classifier, you can see that the AUC, the precision, and the recall is all the same, and it doesn't perform very well. And it's encouraging to see that across all of our three test models, for the most part, the cross-validation performances are better than the dummy classifier. Furthermore, if we focus on the colored points, which represent the test set performances, we can see that the test sets perform very well. So this gives us confidence that our machine learning models are predicting structural cardiotoxicity quite well, and that the genes that we selected are reliable. But now we come into the feature importance side of it. So when we look at the important genes which were selected in any one of our machine learning models and add them together, we get a total set of 52 genes, which were highlighted as important in at least one of these models. And so we took these 52 genes forward and we tried to see what they represented in terms of biological pathways and functions. So in order to achieve this, we pulled together information about these genes from different sources. One way was to look at the gene ontology terms that gene was a member of when we performed the enrichment analysis from our WGCNA analysis. Another, of course, was to look at our gene function. We also went back to our differential expression analysis and looked at the significance of association with structural cardiotoxicity. But also we were interested in where these genes were expressed. So we looked at GTEx for heart tissue expression of these genes, as well as human protein atlas to give us an idea of the single cell expression of our genes in cardiomyocytes, endothelial cells, and cardiac fibroblasts. We also performed a literature review to try and see if these genes had been linked to cardiotoxicity before. And based on all of these different sources of data, I then annotated the pathways that these genes could be involved in. Based on all of this work, I think there are two main takeaways from our 52 gene set. First of all, I want to highlight that it's very important to look at non-cardiomyocytes when studying structural cardiotoxicity, as well as cardiomyocytes. And the reason I say this is because many of the genes in our 52 gene set were either more highly expressed in non-cardiomyocytes than cardiomyocytes, or even exclusively expressed in non-cardiomyocyte cells. A second takeaway message is that these 52 genes were putatively assigned to one or more of 13 pathways linked to structural cardiotoxicity. And I think this really highlights the multifaceted nature of structural cardiotoxicity as an endpoint. And it emphasizes the need to going back to what I said earlier, to have lots of different assay parameters covering different aspects of structural cardiotoxicity to really have a comprehensive assessment. So to summarize this part of my project, we identify a transcriptomic fingerprint which captures diverse mechanisms of structural cardiotoxicity. We also show that many of these genes are expressed in non-cardiomyocytes as well as cardiomyocytes. But of course, because this data set is quite small, we want to validate the genes we found in a follow-up transcriptomic study, which includes a larger set of toxins and non-toxins. But in the future, this transcriptomic fingerprint can be linked to phenotypic measures of structural cardiotoxicity to improve safety assessment in early drug discovery. So now we come on to the final part of my talk, which focuses on metabolomic analysis of structural cardiotoxicity. So the power of metabolomics is that it's a very dynamic readout of different perturbations, and therefore it could give us a complementary picture of how our compounds are inducing structural cardiotoxic effects on cardiac microtissues. So just to recap, the metabolomic profiling that we performed was using the same study design as in the transcriptomic dataset. So this really gave us an opportunity for multi-omic integration and analysis as well. And in this study, we did metabolomic profiling on the intracellular metabolome of cardiac microtissues, as well as on the extracellular media. So I'm pleased to show you some initial preliminary results based on this. For the metabolomic integration with transcriptomics, we used a method called multi-omic factor analysis. And this is a figure taken from the paper of that method. So essentially what it does is it's like a principal component analysis performed on multiple different assays. In this case, we have 3 data sets, one for the intracellular metabolome, one for the extracellular metabolome, and one for the transcriptome. And then we can decompose each of these data sets to get weights or importances for each of our different molecules in the data set, as well as different factors, which are like a low dimension representation of the data sets and capturing the variation in the overall data set. So when we look at multi-omic factor analysis, we specify, given the size of our data set, that we would like 12 latent factors to be calculated. And one of the things we need to assess is how much of the variation in each of the data sets do these factors explain. So for example, in this preliminary data we have here, factor 1 explains quite a lot of variation in the transcriptomic data set, but not as much in the intracellular metabolome or the extracellular metabolome. And as we go down the different factors, we can see that generally none of the factors explain a lot of variation across all of the different data sets. So this suggests that the intracellular and extracellular metabolome are capturing different aspects of variation from the transcriptome. Equally, we can look at the total variance explained by all the factors for each data set. So what we can see from this bar chart here is that the latent factors cumulatively explain quite a bit of variation in the transcriptomic data set and quite a bit in the intracellular metabolome as well. However, they explain considerably less in the extracellular metabolome. And moving forward, it would be interesting to see what different covariates these latent factors are correlated with. We can also prioritize those linked to SCT, and we can identify the contributing genes and metabolites in those latent factors, which could be important for SCT assessment in the future. So I've shown you 3 different aspects of ways that we're trying to improve the interpretability and assessment for structural cardiotoxicity. I think, if anything, there are 3 take-home messages from this work. First of all, that SCT is a highly multifaceted outcome, and therefore we need lots of different assays and ways of assessing it to really understand the mechanisms underlying SCT for appropriate de-risking of compounds. Furthermore, I've shown that with the transcriptomic data set we had, and hopefully with the metabolomics as well, we will identify multi-omic signatures, which can provide greater insight into the mechanisms of structural cardiotox. But more importantly, a part which I touched upon but didn't really elaborate on, but it would be good as a future work point, is that we can link the assay endpoints which we identify to multi-omic signatures, and therefore create actionable interpretations to manage high-risk compounds, and ultimately work to reduce attrition rates in early drug discovery while creating safer compounds for patients. So I'd like to finish off by saying this work was by no means done just by myself. There was a very large team of researchers and scientists behind this, and I'd like to extend a warm thank you to all of them for all the help that they've contributed to this project.